Hi, Maggie.
Good morning. Good morning, everyone, and afternoon for some of our folks. I'm Robin Shapiro, and it's wonderful to see so many people joining us today. We are very honored to have Dr. Larry Corey, who is professor of medicine at the UW, among his many other accolades, which I'm sure you've read about, and Dr. Dale Reisner, who is our Health Advocate X board member and medical director of OBGYN quality and safety at Swedish Health Services. So as you know, this is a public recording, and today we're going to be talking about the COVID-19 virus and the impact of vaccines. We're going to dig deeply into the virology and the variants and look forward to having a very robust discussion. As always, if you don't want to be featured on this public recording, please turn your video camera off. And we recommend that you send questions through the chat function, and I'll be moderating this call. So with that, I'd like to turn it over to Dr. Dale Reisner. Dale?
Yes, good morning, everyone. For those of you that are at least on the West Coast, and I guess we're into afternoon for the East Coast folks, welcome. I typically start with data, and I'll do the same today. I have a few comments to make about this morning's release of the CDC guidance for vaccinated folks, and then we'll turn the program over to Dr. Corey and really have the bulk of the program focused on his expertise. So if we look globally, we are now at almost 117 million confirmed cases with 2.6 million deaths. That's global. And in the US, we are at 29 million confirmed cases with 530,000, a little over that, deaths from COVID. Specifically in tested cases. So we are not looking at other types of death that may have been related to lack of care during the pandemic. In Washington State, where we are housed, we have almost 350,000 confirmed cases, and the death rate is a little over 5,000. Our death rate in Washington State is 1.5% of confirmed cases. So when we first started the pandemic a year ago, amazingly, we were thinking the death rate may be as high as 5%, but that was really due to biased information and the lack of full ascertainment. And even now, we're not testing absolutely everybody. So we may be closer to 1%. I think that is what most of the infectious disease experts are expecting. Knowing that we're, asymptomatic people are not all getting tested, or mildly symptomatic even. So what I wanted to let everybody know is we have had November, December, January, particularly December and January, the highest peaks ever in the country. For cases, we have come down significantly, and most places are plateauing. I just want to remind everybody that the current plateau, so if I just look at King County, where we are mostly located, the rates are now similar to what we had in our peaks in April and July. So we still have a pandemic. It is getting better. The ability to vaccinate is huge. And the message obviously is that we still need to be super careful. We have variants, and I know Dr. Corey will be able to talk to us a little bit about how that impacts vaccinations and so on. But we still need to be really careful. And so don't get too frustrated with slower opening of things that you think you'd like to have opened up again. I will say for the state of Washington, we're really lucky to live in a progressive state, very public health focused. The governor announced that pregnant women will be included in the 1B3 group, which will be as of March 22nd, I believe, and other essential workers that were not included already. And we're really happy in the OB arena to hear this. There's been no data on pregnancy and vaccinations. We feel like these vaccinations will be the same in pregnant women as they would in any other women their age, but there's no specific data. Pfizer's conducting a study currently on that, and we have, our national organizations have encouraged very much shared decision making with provider and the pregnant person to determine whether they get vaccinated or not. But we are encouraging vaccination, so really a good move, I think, on the part of our governor's health team to do that. So I wanted to also just mention, in Washington State, we have had well, in the US, we've had over 58,000 vaccination doses given out. So that's almost 18% of the population has received at least one dose. And it's about 9% who have received the full 2 doses. So it's still a tiny proportion of our overall population, and that really impacts how we act, and just to kind of put it into context. And so with that, I think the other thing I wanted to mention is the CDC guidance. And then I'll turn it over to Dr. Corey. And really the key point, so people who've been vaccinated have been a little frustrated because they don't know what's safe to do and what's not good to do. And we will still continue to say, until we have all experts in the medical field agreeing that the pandemic is under enough control or is done with, that we can really be somewhat back to normal. That if you're fully vaccinated, it should be fine for you to visit with other fully vaccinated people indoors without wearing masks or physical distancing if you're comfortable doing that. You can visit with unvaccinated from a single household. And this really impacts folks that have grandchildren and really want to hang out with their grandkids, have been afraid to do so without wearing masks or physical distancing. Obviously, with young kids, that's really hard. So that is felt to be reasonable. If you are exposed, you find out you've been exposed later, but you're asymptomatic, you do not need to be tested. If you have symptoms, you definitely should go get a COVID test. So if you fully vaccinated and you've been around somebody that says they're now COVID positive, use your symptoms to base whether you need to get tested or not. And obviously, if you're nervous about it, you can get tested, but it's not a recommendation. And it's also not a recommendation to quarantine. So those are the key points. And if there's questions about that, happy to answer those towards the end of the talk. And I wanted to hand it over to Robin and Dr. Corey.
Great. Thanks, Dale. And I know that we already have a few questions coming up on the chat that we'll get to after we spend a few minutes with Dr. Corey. So Dr. Corey, I thought you could just lead us off with talking a little bit about the mRNA technology. We haven't really focused on that in our previous community conversations. So talking a little bit about why people should feel confident about that technology. It is new, and the difference between the mRNA vaccines and the J&J vaccine that was just approved.
All right. Well, I thought I'd sort of start a little bit more at a sort of 30,000 foot level, because it's about, I think a year and a day since I sort of wrote my first emails about starting the country's COVID vaccine program. I sort of came back from a HIV vaccine meeting. I run the HIV Vaccine Trials Network, and I founded that 20 years ago. And people need to know that I've worked with a fellow by the name of Tony Fauci for 35 years, and he's really one of my close personal friends. And we eat dinner together at a place in Washington, D.C. every 6 weeks for the last 10 years. That's half friends and a lot of business about basically the HIV vaccine program. And early in March, he called me and said, "Hey, Larry, this is, we had already talked when the virus was discovered, this virus had traction. For me, it's still interesting that it has become very early on so human adapted, and to my knowledge is, if it jumped from animals to humans, which it sort of has, and there's evidence of that, it is the most adapted human virus. You know, you could tell right away from Wuhan that it wasn't SARS-1, it wasn't MERS, that it burned out after a while because it's not uniquely adapted, and that this was going to have, quote, traction. So it became apparent that the tool that was needed was a vaccine, and it was really one of developing the infrastructure to test and use vaccines. And science builds on science, and all aspects of what happened in this miraculous 10 months from starting to getting these incredibly effective vaccines. This is some of it is luck, a lot of it is the prepared mind, but it is science building on science. The construct that Barney Graham and the Vaccine Research Center developed is really, the Vaccine Research Center 20 years ago was developed for an HIV vaccine and went into, emerging infections and pandemic infections with influenza, branched out a little bit into RSV, and sort of the understanding that you needed to, the best neutralizing antibodies for when the virus attaches is in the open frame, in the stabilized open position, and that was found out from RSV. When a virus lands, it lands like this, and then it attaches, it closes like this. And if you just hold your hands up and you go like this, it takes a lot more energy to keep your hands open than it does here. And so those in order to, when you were making a protein, you needed to stabilize it, and stabilizing it meant you had to really stabilize it so it wouldn't collapse and fall together. And they had made some vaccines that failed in RSV, and they actually found that they made the protein and would collapse in the bottle. And that's what was good originally did not turn out good when it went into people's arms. And out of that failure, they learned how to stabilize in the open fusion frame. And within 3 weeks of the genome being posted from China, they had developed a stabilized spike protein RNA sequence that actually looked like it could be stabilized and could be expressed in a stable form. So that history was sort of one that occurred, and actually that piece of RNA is sort of the same piece of RNA in the Moderna vaccine and the Pfizer vaccine. So they each have their encasing in soap bubbles, we call them lipid nanoparticles, but they're microscopic soap bubbles that protect the RNA from the RNases that are in our tissues and in our blood. So that's one aspect of science building on science, and the other aspect is the infrastructure that was created in the clinical infrastructure to test to test vaccines have been built in the HIV system. And we mobilized the country's academic infectious disease programs throughout the country to sort of participate, and formed what we call the COVID vaccine trials network. And we started working on that in April and May. Then the issue was how to do this, and how do you conceptualize doing this? Now, the U.S. government, and this is the Trump administration, so did want a vaccine development for SARS-CoV-2, and put a significant amount of money given to BARDA, which worked with NIH into developing a sort of U.S. government portfolio for vaccines. And that was, essentially reasonably thought out. Everybody knew that you would need more than one vaccine. That right away became apparent that all the adults need to be vaccinated. So that's 220 million people in our country. Became fairly clear that Europe that the globe needed to be vaccinated, and that's 7.7 billion people, about 5 billion adults and 3 billion children approximately. So there's no one technology that was going to be capable of doing that. Second aspect of that was, okay, let's use the platforms that we know make vaccines. And we talk about, I'll classify them as genetic immunization, proteins that mostly are the proteins that you make that we take in the vaccines, the hepatitis B vaccines, the HPV vaccines, the norovirus vaccines. They're all making proteins that are usually the outer coat of viruses to which you make antibodies, and you therefore recognize them and bring back memory if you get exposed. You know, you make immune responses, and that's how vaccines work. And then the third technology is the viral vector vaccines, in which you sort of use a virus to actually carry the vaccine in, the piece of DNA in, and so we'll compare and contrast the RNA with the virus. But you can think of the virus, both RNA and whether it's a lipid soap bubble or a virus, they are just shuttle vectors to get that piece of genetic material into the cell and let the cell do its work by transcribing it. And that's where these technologies are all about. So they picked two platforms for each sort of type. Pfizer and Moderna ended up, making the RNA. The chimp AdOx, or AZ vaccine in the United States, and the Ad26 vaccine are the sort of the viral vector vaccines. And then the two proteins, one of them is made by Novavax, and the other one is made by Sanofi Pasteur. So there were essentially 6 vaccines originally, and the Merck started doing one live virus vaccine, and that hasn't worked out, and they've left the field. But so it was a rational decision, and the second decision was to work with large pharma, because they were international, and B, it was scalable. As sort of for us as scientists, we always viewed this as a global program, and despite all the America First stuff, and it's designed that way and was designed that way, and the J&J study is an overt manifestation of how that was designed, and that was designed way back in April and May. We essentially designed these trials. So,
and I want to jump in here, because I think one of the things that's been so surprising, I remember kind of midway through that people said that we were going to get a vaccine within a year, and no one thought that was possible. So how could that possibly happen, that the vaccine was able to be brought to the world so very quickly, and people have, great confidence in it?
Well, I think the issue is you learn from this experience that when everybody is affected and everybody wants to solve a problem, you can work together, taking academia, industry, and government. If you take money out of it, and huge amounts of money were put into de-risking every aspect of this, as well as trying to take away all the administrative issues. For us, and working in, I started this work before Operation Warp Speed was, quote, created, and we designed these trials essentially at the hutch in my group with the statisticians, and even before Operation Warp Speed was sort of officially started. And but when you worked in Operation Warp Speed and you had a call, it wasn't, let's meet again next week. The operant issue was if you couldn't meet tomorrow, then you had to meet in 48 hours. There was no phone call that wasn't the follow-up, wasn't within 48 hours. And you worked 7 days a week. And it didn't really matter, this is what you did. And that's sort of what has been done, and hundreds of people work that way, including the companies, as well as manufacturing facilities were commandeered, basically, and all sorts of dollars were put into things, and you designed phase 1 studies to go to phase 2 studies and phase 3 studies. So you didn't wait to normally you would wait and you'd make an economic assessment, then you go to phase 2, and then you wait an economic assessment. the studies were done just as you would do any other studies, and the regulators and the FDA were really wonderful colleagues. So the phase 1 studies were a couple hundred people, the phase 2 studies were 700 people, the phase 3 studies were huge. I mean, when we started them, the statisticians said that 12 to 15,000 people was sort of what we might need, and I basically made the decision to double them. Because time was money, and the sooner we got the answer, the more important, the more time we would have, and that's where the 30,000 number came from.
And I
So,
think that that's very important, especially for all the advocates out there who are working with patients and might be talking with them, their friends and neighbors, about people who are not confident or have some kind of myth about the vaccines, that the vaccine process, development process, which went at really warp speed, followed not the same process, but as rigorous a process as normal drug development goes through. I think that's important for people to understand. Mm-hmm.
And the results were sort of remarkable. Pfizer didn't want to play. They sort of didn't want to play with the US government. They wanted to use their own thing, so that's fine. But here's their study at 43,000 per person, and the one that was in the US government, the OWS program, was Moderna, that was 31,000. So these are very large studies with the same strand of RNA with a little bit different soap bubble. And the results are identical. I mean, I don't think I could have ever even written a book on the fact that you could independently do 2 independent clinical trials in the 40,000s. They inject the same piece of RNA into your arm, and the transcriptional pattern, the antibody pattern, and the results, the clinical results, are essentially identical. They are identical. And that is sort of like, huh? That is, never would have predicted that.
Yeah.
And so, what you're doing here is, and what we're learning, and why I think RNA vaccines are going to be, have traction is in the future, is that you're basically learning that the cell's ability to transcribe a protein is, and do it and put it in the right conformation, is, well, let's use what we have, what God made.
Mm-hmm.
If you look at the timeline, RNA started first, came off first. The viral vectors started second, came off second. And the proteins, which are just harder to make. And once you make them, they're cheap, it's great. But, Sanofi Pasteur failed in making its first protein, and they just, and lost 4 months, and they're still starting their phase one trials were just enrolled in the United States and Honduras today, and maybe we'll get in May that protein vaccine, and it's going to be tough to even test it against placebo, and so for us, the United States, it's not even going to be relevant. For us, it'll be useful for the world, but it just shows that these technologies do make a difference. You can get into the clinic and you can do this. The way science is done, you have a piece of genetic material, you paper clone it, you take the code and you send it to a company, you pay them $150, and 48 hours later by FedEx, you get the strand of DNA back and you start working on it. I mean, that's sort of, that is the way science is done. And every lab in the country does it just this way.
Yeah. Let me jump in here. We are getting some questions in the chat, and I wanted to make sure that we addressed this idea of the J&J vaccine. And my understanding from reading your blog that's posted on Johns Hopkins was that the J&J vaccine has 82 to 90% protection against severe disease, but the whole statistics around J&J and how effective and that kind of thing has kind of been, I think, misunderstood in the public. Can you address how effective the J&J vaccine is, how it's different from mRNA vaccines, and what's the most important things for the public to know about J&J?
Well, first of all, the strand, the spike protein that is essentially the same in J&J and the RNA. It has a little bit of a difference in that it sticks to the membrane better, but so it's a little bit longer in its length. But how it's delivered makes a difference. When you put it into the viral vector, the expression of T cells is probably, especially cytotoxic T cells, is better with the viral vectors than it is with RNA. But antibody is a lot higher with the RNA than it is with the J&J. And so, the issue
it
is
works a little differently.
So it works differently. The issue is, what's the goal of what a vaccine should do? When we designed the trials, everybody felt that the most important thing was to prevent disease and death. Still, it is the most important thing. And that's the trials were designed to find out, did it ameliorate disease, and did it prevent severe disease, and did it prevent the complications of the disease? And we designed the J&J study to be an international study, and there were two reasons for that. Number one is that we had worked together in HIV, and they had actually had an international focus, and they were a global company. We actually didn't know that the variant would appear in South Africa or Brazil. I actually used it as an international study because I was worried that we didn't have the capacity to do 6 clinical trials at that time of 30,000 in the United States. And I thought offloading some work to international sites where we really had a lot of good people was a good thing. And it turned out that we were prescient about that. You know, it turns out that we now have data on the variant. So the real issue is that there are two reasons why the efficacy, overall efficacy in the United States was 73%, not 95%. That's probably because the antibody levels are not as high with the 1-dose J&J vaccine as the Moderna vaccine. There's no question, actually, the Moderna vaccine gives you higher antibody levels after the second dose, 50 times higher. Titer is like 450. With the J&J vaccine, it'll be 40 or 50. Okay, so it's really very different. But why
didn't they study a second dose with J&J?
Well, sometimes politics win. We wanted it to be a 3-arm study. Let's just say they're politics. They did a second study of 2 doses. We had learned in the HIV system that to administer the second dose after Ad26, you had to wait 2 months. It wasn't very good if you waited 1 month. That meant the study was even a month longer. There was this desire of having speed. And so it was a desire to separate the study rather than to do it all in one study. Hindsight's 2020 was probably not the right idea in the first place. So I won't relive that one, but they do have a two-dose study. I'm sure it'll probably be a better. I'll be shocked if it doesn't have a better response. It's almost fully enrolled, and we'll find out about it in a couple months.
Okay.
I think most people who are getting 1-dose Ad26 will get another booster. You know, a year from now, there'll be a recommendation to have a booster.
And we had this strange, well, I call it a strange question, just because I haven't seen it before, but Seema Khan says, word is that the state of Virginia is advising those who had Moderna as their first vaccine to get a second shot J&J. Is this valid scientifically? I hadn't seen that before.
It probably is valid scientifically. It's a data-free zone. I actually don't think that that's what the state of Virginia should be saying. But it's actually, you know, I guess, you know, if you really want, from an immunologist point of view, I'd prime with J&J and boost with Moderna rather than the other way around. But, you know, that's I think in the end, we will do some mix and matching that'll probably be mainly for immunosuppressed people. I don't think that for the immunocompetent people, we need to necessarily do that. Maybe eventually we will get around to that. The companies don't like to cooperate at the moment. These are lot of development. There's large markets. This is where the clinical trials were done. We did that on purpose, not to do mix and matching, because we needed speed and we needed their cooperation, and we needed, this is, and it's amazingly as to how well it did happen. So I don't think you should mix and match things at the moment. There are some studies that are starting to look at that. And eventually there's no immunological reason why it should do any harm. But I think you got to follow the data, and the data are, the 2 doses of Moderna, 2 doses of Pfizer, incredibly successful. The 1 dose of J&J, incredibly successful for preventing you from being in the hospital, from getting on a ventilator, from dying, has known protection against the variants we're talking about, both the Brazil variant and the South Africa variant. It will prevent you from dying. And so that is actually, when you look at the variants that are coming out, we have more uncertainty with the RNA vaccines than we actually do with the J&J vaccine.
Mm-hmm. Well, and we do have questions about whether or not we'll be needing to have booster shots in lots of questions coming through. But so will this be like a flu vaccine that we need to get regular vaccines annually, or do we know? Or
well, I have to say, at this point in time, there's an enormous amount of things we don't know. And I think you just have to say, at times it's a great problem to have, that we actually have this miraculously effective vaccine that, yes, we developed in 10 months, and does it prevent acquisition? Does it prevent, can you get colonized in your nose and not know that you have it and still transmit it?
mm-hmm.
What's the reduction in transmission and contacts and transmissibility with these vaccines? And the answer is we don't know that. What we do know is that it'll protect you from getting bad disease and from dying. I mean, there's
so can I just follow up on that to make sure that I understand? So what you're saying is that we don't yet know if you've had the vaccine, you could still maybe get COVID or pass it along to someone else. We just don't know.
You could we don't know whether you can asymptomatically get infected and pass it along to someone else. Now, I suspect that that will not be common. I suspect that it may be more common with the variants than it did with the Washington strain that's currently, that had been circulating, because the UK variant and the South African variant have some genetic mutations that really do increase the transmissibility significantly. And so maybe with a more infectious virus, you will get colonization in your nose and not know it, because you are going to have an immune response, but yet are you still infectious to others? And until we get higher vaccine coverage, it's sort of like measles. Think about it as measles. I mean, to me, the whole concept of herd immunity has actually been both overplayed and ill understood, and is, I'll only say, is irrelevant with this virus, because this virus has actually already shown that it can escape from natural infection.
So can you maybe simplify that, or go a little deeper on that one? Yeah.
So the idea of herd immunity is basically a little bit of a weird one, to sort of say someone else gets infected or gets protected, and because so many other people are protected, I'm unlikely to get infected. And people have been walking around sort of like this attitude of, gee, let everybody else get infected or vaccinated, and if I'm a little bit hesitant, I'm going to be fine, because I'm going to, the virus will not actually get to me because everybody else is protected. Well, there are a lot of viruses that don't operate that way. Measles is one of them. And the reality is this virus is telling us it doesn't operate that way. So first of all, we have very good data from South Africa that people who got infected from the European variant that was first circulating there, let's say from January to September, when the new variant appeared, which has multiple resistance genes, it's like 14 or 16 different mutations that are different than the original Wuhan or Washington strain. Those people actually still got symptomatic disease. Now, it's not clear whether any of them got, death or dying, whether that prior infection prevented you from dying, but it certainly didn't prevent you from getting symptomatic disease. We would therefore call that an escape variant, because here you got infected and, it escaped, similar to like influenza, except more frequent and worse because we've had 20 influenza infections in our life as an adult as you get reinfected with a new strain. Here's just, you're getting a new virus into the human system. So it is the concept that prior infection is going to help you from getting other people infected and transmitting it is not, has already been shown. The virus can under pressure. In fact, you'd ask the question, why is this, why do the escape variants are so prominent in Brazil or South Africa or, and not in the United States where we've had more genetic stability? And the reason is we have a huge population that has not yet been infected. Even in Seattle, where we started with this epidemic, we have only 10 or 15% of the population infected. I mean, this virus has a lot of people that it has the chance to get into.
So there was
we're very far away from herd immunity.
Yeah, there was an editorial actually in today's Seattle Times about just that even if we've been picture perfect on locking down, that means that actually the virus has the opportunity to basically get in there.
What is relevant is vaccine coverage.
Yeah.
That is what the relevant issue is, because we actually now know vaccines actually give higher rates of immunity than natural infection.
Yeah. So get the vaccine, take whatever one's offered. That's the big thing. So Ray Dresden has been very patient. She asked her question a long time ago about vaccines and vaccine trials in children. So can you talk about when that might be coming? And
well, it's partially started. I think the deficiency we're having and spending a lot of time on and trying to deal with it is the elementary school kids. So Pfizer, has sort of took it down to 16, and now they're almost finishing their study between 12 and 17. Moderna just fully enrolled their adolescent trial, and we'll hear more about that. And we're working on J&J to move their platform along, because actually their vaccine for Ebola is licensed by the WHO for both pregnant women and kids. And so there's an experience with that platform with both kids and pregnant women. So the regulators are just starting to the FDA is just starting to sort of try and define the criteria. No one's planning on doing efficacy trials. The real issue is what's the right dose and what's the safety profile. Now, 12 to 17-year-olds, everybody's sort of using mostly the same adult dose, and I think it's going to be fairly easy to get to that. The lift, I think, and where I think women want this to be, and I'm with them. I have two daughters-in-laws who have lots of grandchildren who are in elementary school, and everybody wants their kids back. It's been, how do I do this for the 5 to 12-year-olds? What's the right dose? The RNA second dose is fairly reactogenic, and so you probably need to lower the dose in kids, and you will get they tend to have a better immune response anyway. So you got to find the dose and you got to find the safety, and that takes a little bit longer. Personally, I don't know why it's taking so long, but I am pushing so that let's try and do this by the end of July or August. There's an official white paper to the president that basically says, don't expect this to the first quarter of 2022. Let's just say there are a lot of people who are in, quote, the operation now, including Dr. Kessler, who's the chief scientific officer, and certainly Tony, that are really sort of saying, wait a minute, we should be able to do this quicker. So we'll see if we can deliver on that. But it's certainly something we're trying to do.
So we have a question here from Kathy Bowers. Please comment on the CDC, PIC, pneumonia, influenza, and COVID being lumped influenza together with COVID. Did the CDC confirm that only 6% of COVID-related death certificates indicate COVID as the only cause, or 94% list other illnesses as the cause as well? So if you get COVID,
Well,
you're more
I guess I
likely
have to sort of answer that there is this thing called excess mortality. And it's been going on it's actually what I worked on 40 years ago when I was at the CDC. It got me into infectious disease, and it was in the influenza branch. And mortality rates in the United States are age adjusted, and they are like clockwork as to where they are, and varied by science, and it's been like that for 70 years. There are only two things that have ever, prior to COVID, raised excess mortality rates in the United States, and that's been influenza and heat waves in the Midwest. Neither one of them are occurring now, and there's an enormous increase in excess mortality. And so to think that, and this is a systemic disease, people are dying of pulmonary emboli. I mean, this is not flu.
Yeah.
And this is a bad disease. Okay, it's a multi-system disease. If you actually look early, there's viremia. I personally think that most people have viremia. There are viruses that actually travel in the lymphatics and are cell associated, but you find it in the heart, you find it in the spleen, you find it, in autopsy material. They have kidney disease, they have endotheliitis, which is, in micropulmonary emboli, and some people have myocarditis, but, they get AFib. This is not, for anybody to think that that this is not killing people is incredibly medically naive.
Yeah. So, and to that effect, there's a question about studying the long-term effects of both the virus and the vaccines. And so can you talk a bit about that?
Well, I hate this term long haulers or whatever. It seems disrespectful, but chronic infections and chronic persistent symptoms, including anosmia.
What's anosmia?
That not smelling and not losing your sense of taste, does seem to recur in people. And I look at the diseases, you can look at it and saying 100 million people have this, and yet we know so little about it. And we're learning as you go forward. And one of the issues is when you are in a pandemic and you're so overwhelmed medically that you don't actually have time to think about what the research questions are and how do I do this. So I think we don't understand. There is emerging evidence that some aspects of the virus will persist, that you can get persistent antigen. It can be retained. We know antigens can be retained in the lymph system. That's one of the reasons you have memory B cells, is that it persists for long periods of time. You persist to have long-acting plasma cells so that there's retained antigen, and maybe there'll be immune response. There's cross-reactive antibodies. There's an autoimmune phenomenon. I think there are a lot of things that are going on that we don't actually understand at this point in time. And I think it will take us a while to look at that. And, you expect that if you got vaccinated, you wouldn't see these long-term effects, or you wouldn't see adverse events with reinfections. I do have to say that, some cohort studies, sometimes you find biases, are showing, a very significant percentage of people with long-term persistent effects. And so, again, that leads me to say, vaccinate. Who wants to have an illness that most people say take you 6 or 8 weeks to recover, even if you're mildly symptomatic, and 20 or 30% of you will, continue to be dysfunctional for months.
Yeah, it's terrible. Here's a question from Michael Wood. Dr. Eric Topol on a recent WebMD webcast said the UK variant is the most threatening. What might we know? When might we know if any of the vaccines are effective at preventing severe disease?
Well, I disagree with Eric.
Okay.
The UK variant is the most, is very infectious, and there's this article out in Science to confirmed that is 50 to 60% more transmissible. It is taking over all of Europe. It is to be a variant that seems to have increased transmissibility. It is a variant that seems to be shed for a longer period of time and hence more transmissible. And I think it is so, quote, going to come to America. But it does not at the moment, the classic UK variant does not at the moment have these mutations in the receptor binding site that make it neutralization resistant. It reduces the neutralizing antibodies required to reduce that are only 2 to 3-fold less. So if you're at 400, you'll go to 200. And that's plenty above the threshold to prevent you from infection. So I do think it may still be transmissible, more transmissible with the vaccine. I think we're going to find that out in the studies that we're just starting, the transmission studies. But the South African variant, the 315 variant, is by far the one that worries all of us the most, because that requires 8 times more and neutralizing antibodies to transmit. It has multiple genes that that are escape resistant neutralization, as well as having the mutation that's associated with increased transmission. And South Africa had this enormous second wave and showed that with a really steep curve, showing that it was really very transmissible. And then this virus is more neutralization resistant. And that's the one most of us are concerned with respect to why the South African variant is entering into clinical trials. It may be, in my opinion, if it's the UK variant, we'll handle it pretty well. And you could handle it with, if our immunity wanes, the same vaccine that we now have just needs to be boosted, and we'll handle it. And I feel very comfortable to sort of say, yeah, if the South Africa variant comes to here, we're going to be sort of okay for a while with respect to the immunity. I may regret those words, but by in vitro testing, that's what you would expect.
Mm-hmm. Okay, so we have a very practical question here from Anne Llewellyn, who, and I believe that she's already had the vaccination. Would you take a vacation to Norway on May 22nd? Small cruise ship, 900 people, thoughts?
I would never go on a cruise ship. I wouldn't have gone on a cruise ship before, yes, but I certainly wouldn't go on a cruise ship now.
But maybe broadening that a lot of people are very anxious to travel and be with other people. So what do you see happening in the near term this summer in terms of our ability to be in larger crowds if we've been vaccinated? What do you think about that?
Well, at this point in time, I'd like to know, there are a couple things that I say this about the cruise ship is, number one is, I'd be pretty comfortable that I'll probably be relatively protected. But I also am going into a situation at this point in time where still in Europe, they don't have a lot of vaccination. And you're having a lot of disease. You have different variants in Europe than we have at the moment. They have way more of the UK variant. And even though, but the point is that there's this other thing called convergent evolution, in which the more you have people infected, the more viruses you have, the more the other variants can come out. So the likelihood of the UK variant being like the South African variant, and acquiring these E to K mutations and some of the NTD mutations is still very high, because Europe is not doing very well. I don't know exactly about Norway, it's a wealthy country. Are they all going to be there? But on a cruise ship to Norway, you're going to get a lot of people from a lot of different places. And actually, I don't like that situation because it's an incubator and you're in a closed ship, and it is a documented high attack rate place. So if I was, if you said I wanted to take a walking tour with a group of 6 people in Norway, I'd probably say, sei gesund, go do this. But
and I know what that means.
but on a cruise ship, I don't know.
So basically what you're saying is
I'm not your travel doctor on the cruise ship.
yeah. So basically what you're saying, and Anne just said it was for 2022, but basically what you're saying from a very practical level is that while we will be able to do more things than we've been able to do for the last year, don't go hog wild and be, thinking that we'll be in large crowds and, enclosed spaces in large crowds, probably in the near future.
No,
Right.
I think it's until we get full vaccine coverage and then we actually sort
Mhm.
of see that how this makes the virus behave. The first
Mhm.
action of infectious disease is never underestimate your pathogen. And I think we've all underestimated this pathogen from the very first time. Okay, we looked at Wuhan, we saw what was happening, and we did nothing in the month of February. And it's not just we, the United States, it's the entire globe. And this virus seeded itself throughout the entire globe in the month of February. And we're paying for it since. And it's been a stochastic issue because we had millions of people here. There's no way you're going to contact trace yourself out of this.
Yeah.
And the only way you do this is by vaccination. And using the tools of modern virology. And it's going to take a while to get everybody vaccinated. So I think the United States will be less and less insular as we move forward with vaccination. I'm seeing my grandchildren now. One of them is in preschool, and the other one is in whatever kind of school you want to call that they do at the moment, spend more time getting there and more time wiping the school down than they do teaching. But I feel comfortable with them.
Yeah.
But yeah, I'm not going to indoor restaurants at the moment.
Yeah.
I'll eat outside and sort of things like that. So,
Yeah. And I'll just add, the CDC specifically says avoid medium and large gatherings. So again, we are on the verge of making progress. And I think the whole combination of the vaccination with the same message, whatever vaccine you're offered, take it whenever you can, as soon as you can. Follow up. If it's a 2-dose, make sure you follow up appropriately. And really be careful, because we know from our own history what were super spreaders. There were weddings, which were outdoor, but close contact, and there were churches, and there were all kinds of places where there were large gatherings, and restaurants and bars were, even with smaller places, spreader places. So I think people have to be careful and take your vacation, but go to the coast, or go with your family someplace where you can rent a house somewhere. Be practical and a little bit cautious for quite a while.
So we have this question, it's a good one. Assuming that the vaccines for children won't be widely available until 2022 or longer, but most other older adults, including at-risk teachers, will have the chance to get the vaccine by August. How does this affect the public school landscape this fall? What percentage of general population educators in the school have to be vaccinated to determine if it would be okay to return to regular schoolwork? And I think some schools are already opening up.
well, I'm probably not going to be a fan of the teachers union here. But I think that teachers should be vaccinated, and I think once they're vaccinated and the adults are vaccinated in the school, then it becomes the issue. What's the relative risk to the children? Certainly, the country wants to have the children vaccinated, and I think we should, and that is, yes, the frequency of, and I would say the pediatricians I work with are incredible advocates of kids. There's articles that actually sort of say, well, elementary kids don't really infect adults very well. Certainly middle school and high school kids socialize more, and they can affect adults more. So I think the consensus in the United States is, because school districts don't like relative risks, it has to almost be an absolute risk, of zero. So I think the consensus of our country is everybody needs to be vaccinated, and we'll all feel comfortable about, having school sort of reasonably back to normal. And I buy onto that, and I think that's probably where it is. Could you, some schools might make the issue, or some parents would make the issue that if the teacher's vaccinated, I think is better off for my kid to be in school, and I'm worth the relative risk to the child as it relates to COVID complications. But the reality is that there's no absolute reduction to say that no kid will not get a complication and have a problem with COVID. That obviously can occur. There are some syndromes associated with immunopathology, and you do see an occasional child who has some immune deficit that you didn't recognize. So anyway, it's a long-winded way to say, yes, let's vaccinate everybody and get back to school. And I'm an advocate of that.
And for kids to be vaccinated, we're looking at how long? Like a year out, 6 months? What do you think?
Well, I'm hoping we can push it so that by Labor Day, you can have the data to do that. Of course, vaccinating it takes a while. I don't necessarily view that what we're doing at the moment is the most efficient way to vaccinate people. Opening up the school and vaccinating the community is what we did with polio, and what we did with smallpox. And there are other ways of doing mass vaccination to let things happen. But maybe we'll be creative about that. And when it comes to kids in schools, we'll use the schools to vaccinate all the kids as we did in polio, and we'll get it done efficiently so that everybody can get going.
Yeah, just to add, with kids getting back to school, there's a huge equity concern for the benefits of school for children in low-income settings to have more than their education from there. That might include a hot meal, some protein, and interaction that they can't get in their homes. Many of the folks on the call are well educated and can supplement education at home, and these kids don't get it. So I would fully support anything that it takes to get safe situation for kids to get back to school from that perspective as well.
We have one last question. We might be able to take one or two more. We're just a couple minutes before the end, but this one comes from Bonnie Sharon. I use the adjuvant, aka kicker sequelae, that causes most of the symptoms after vaccination, not the viral particles. I come from a patient ed background and haven't been able to figure out the makeup of these vaccines. So can you comment on that?
Well, it's like the use of the English language. The lipid nanoparticle that encases the RNA may have some antigenicity. And it also apparently has some adjuvant effect also, because it leads to the dissemination of the particle throughout the liver and the spleen, and there's a lot of antigen processing cells, and that may be one of the reasons why RNA is so effective, is that there is the lipid nanoparticle. The virus, Ad26 in the Johnson & Johnson, is an antigen. It is a human adenovirus that is not that common in the West, but is seen more in the East or in the lower and middle incomes. And yes, you make immune responses to that, and that can be both good and bad. But it does have an antigenicity that brings in accessory cells and inflammatory cells, and that can add to the adjuvant effect as to why with one dose you get pretty decent responses. Classic adjuvants are with the protein vaccines, and both of them, the Novavax vaccine has a matrix adjuvant, and the GSK Sanofi vaccine essentially uses the adjuvant that's in the Zoster vaccine. So, it's a long haul in getting a new adjuvant approved. The Novavax vaccine, this will be the first approval of what they call their ISCOM adjuvant. The GSK Sanofi vaccine, if it gets approved and when it gets approved, will essentially be using an adjuvant that has been used in influenza as well as in adults in the Shingrix vaccine.
Well, and with that, I just want to thank you so much for spending the hour with us, Dr. Larry Corey, Professor of Medicine at the UW, and Dr. Dale Reisner from Swedish Health Services. We really appreciate you giving us a lot of information and a lot to chew on as well, because we don't have all the answers yet, but it was really, really helpful to kind of dig into these variants and the differences between the vaccines. So thank you very, very much. And thanks for all you're doing to help fight this virus.
All right, it's a pleasure to be with you. And I enjoyed it and hope you enjoyed it. Thank you. Bye-bye.
Thank you.